out with a couple of announcements, and then I'm going to come by and just check to make sure everybody's got the homework as we discuss for a little bit. So one, the homework for Tuesday night, so going into Wednesday's lecture, is another group wiki. I just noticed it says, it might say this on some of them, lesson 3, this should be 2. And what we're going to get into starting on Wednesday is talking about the structure of what we call the cell envelope. So this includes outer membranes, inner membranes, cell wall, and then additional features. Really what we're going to be doing is contrasting gram-positive and gram-negative cells, as well as what we call acid-fast bacteria. They don't fit really into either category all that well. Wait, oh, I see why, because this is not the right lesson at all. Here we go. That's the one that we want. My apologies. There we go. It's at the bottom of this list because it's in alphabetical order. Okay. There are 3 columns, 4 rows. Each member of the group take one of the rows. So for example, Annie might take sporulation. Do gram-positive bacteria sporulate? Yes or no? Same thing for gram-negative. Same thing for acid-fast. Double check each other's work on this. Okay? I'll put up a master key, but double check each other's work before coming into class on Wednesday. We actually will start this conversation tomorrow in lab. So our main goal tomorrow in lab is to perform a series of gram stains on different types of bacteria and to begin talking about what is the difference between a gram-positive and a gram-negative organism. In class on Wednesday then, beyond going just the discussion of what is the comparison between the two, we're going to start talking about what effects does that have on the organism in terms of the environment that they live in. For example, why are gram-positive bacteria particularly sensitive to the enzyme lysozyme, but gram-negative bacteria are not? Okay, so that's where we're going with this. So take the time to do that. Let's see. Okay, so here's what we're going to do. Oh, no, one more thing I need to point out. Weekly review. I don't know if you saw my announcement or not, but we're going to start doing weekly reviews in here this coming week. And the reviews are going to be based on a section of lectures. Okay, so for example, there are some fortunate or unfortunate. You can view it as you want, because you only have to do this one time. So let's see. So Jennifer and Paige, you guys will be up for this first one on microbial structure and function. So what I need you to do, and the instructions are on the first page of this, is to come up with a slide for each lecture to highlight one of the major points. Okay? And then to come up with one more slide with 3 questions on it, just reviewing each of the lectures. Okay? I've posted an example, but the idea behind this is to kind of refocus our attention at the end of each of these sections into what's the most important takeaway points. I'll grade these and more so put in any additional comments or notes if I feel like there needs to be something put in there. This is a 5-point assignment. You only do it once throughout the semester. Okay, so this is available for you to look at. Some of the groups are bigger, some are smaller, just based on how many lectures are being combined into that review. Okay? And we'll do these on VoiceThread. And I'll be handing out some instructions on how to access VoiceThread tomorrow in lab. I had a few people test it for me, and it seems to be working, at least with the instructions. So, all right. So here's what we're going to do. We're going to take about 5 minutes. I want you to grab, if you have your table of cellular components on your computer, or if you have a hard copy, bring it up on your computer, grab the hard copy. And I want you to talk with somebody that you did not do this homework with. Because I'm not going to believe that everybody did this individually. And that's okay. I didn't tell you you had to. Well, good. I'm sure some people did, but I didn't tell you you had to. So what I want you to do is check your responses with somebody else. Take about 5 minutes and just back and forth, A through whichever the last letter is. Check your terminology, check your descriptions. I'm going to come by and check to make sure folks have done this. Okay, so just make sure that I can see it as I walk by. Plants that have all plants have cell walls. Fungi have cell walls, but those cell walls are made out of different material. Plant cell walls, cellulose. It's primarily a carbohydrate. In the case of bacterial cell walls, and this is where we'll go on our discussion on Wednesday, the peptidoglycan is made up of peptido. We talked about peptide bonds, so what does this mean? If we break apart the word, which macromolecule are we referring to when we talk about peptido? Amino acids or proteins, right? And then we've got glycan, which is a sugar. So peptidoglycan, there are repeating units of sugar and short peptide chains that create the structure of the bacterial cell wall. Okay. So that's what H is. Other questions? What is F? Is the nucleoid. Anybody put down nucleus? That's okay if you did, but don't ever do it again. All right. No nuclei. So we have a nucleoid. A nucleoid is just a region where the genomic material is located. That's how we define it. Again, no membrane-bound organelles, so there's no true nucleus in prokaryotes, but they still do need to carry out processes of replication and transcription. So that's where the nucleoid is. B here is a plasmid. How does this differ from the chromosome? Go ahead, Annie, get us started.
It replicates independently.
Okay, so the two of them can replicate independently of one another. In other words, when the chromosome is replicating, that doesn't mean a plasmid has to be replicating at exactly the same time. What are other features? Grace?
It has like extra genes.
Yeah, I kind of liken it to like, so I have a 2013 Hyundai Accent. It is a really cheap car. Okay, it cost me like $14,000 when I bought it, brand new. All right, that's the chromosome, is the car. Now, if I started investing money, like if I wanted a really good sound system, which I do not have, if I wanted like snow tires, which I do not have, if I wanted, I don't know, a spoiler, which would just look ridiculous on a Hyundai Accent hatchback, but nonetheless, all of those are upgrades, right? That's what a plasmid is. It's an upgrade in some ways. Plasmids that we will talk about later on in the semester are ones that contain resistance genes or virulence genes. Resistance, antibiotic resistance. So ones that give the organism an ability that maybe it didn't have before. And virulence, we mean the ability to cause disease. In terms of plasmids, they contain non-essential genes. So the chromosome contains, the majority of the genes are essential, ones that are required for this organism to live in the environment that it's typically found in, or environments that it's typically found in. Plasmids contain additional genes that if the plasmid is lost, it may lose the ability to perform some functions, but it can still live in that environment. Okay? So that's the major distinction there. How about, what's the difference between E and A? E is what? I think I wrote this one down because I wanted to make sure there was no confusion with it. Nobody wants to say it because it's a hard word. Is that right? Fimbriae. Okay? Fimbriae is the word there. And then A. What's A? Flagella. All right? Or flagellum if it's just one. And we'll talk about that structure a little bit more today. Fimbriae, required for attachment, or used for attachment to surfaces. There is another structure that the book refers to, and that we'll come back to later on as well, known as a pilus or a pili, P-L or P-I-L-I, or P-I-L-U-S. That's another structure that can be used for attachment as well, but we're going to talk about the pilus in the context of conjugation, which is a process in which bacteria can share genomic information between two cells. Okay? But Fimbriae primarily used for attachment purposes. So the attachment to these cells inside your gastrointestinal tract or to this table right here. Both of those can help for bacteria to colonize or to localize to a particular region. Any other ones that there are questions on? I'm not going to spend the time going through every one of these. Okay, let's go ahead and move ahead then. I'm going to start with, I'm not going to just stick with this picture for a minute, but what's different between this and a eukaryotic cell? We kind of started covering this, but what is different about this prokaryotic cell?
There's no membrane-bound organelles.
That's the first one. No membrane-bound organelles. And probably that's the most important one, or at least the one that we most often distinguish. We've also talked about there's a size difference. We said bacteria, typical size is about a micrometer is a good rule of thumb. There are ones much larger than that. There are some smaller than that, but a micrometer is a good rule of thumb. Eukaryotic cells, if we're talking like human, 10 micrometers in diameter, so about 10 times larger in diameter, which means the volume is much larger than that, somewhere on the order of about 100 times larger. No membrane-bound organelles. Let's go back to that one real quick. I'm going to just write out 3 terms. This process, DNA to RNA to protein, known as what? Let's play around. Central dogma. Right? Central dogma is the process in which DNA is replicated to form more DNA. DNA is transcribed into RNA. RNA is translated into protein. Where do these events take place in a eukaryotic cell? So where does replication take place in a human cell or a yeast cell?
Nucleus.
In the nucleus. How about transcription?
Nucleus.
Still in the nucleus. Translation?
Cytoplasm.
Yeah, generally, right? It could be on an endoplasmic reticulum. It might just be free-floating in the cytoplasm. But bottom line, cytoplasm. Where do these events take place in a bacterial cell? So where does replication take place? Why should we even care where it takes place? Emma, where does replication take place? Take a look back at our picture. You can even use, where is the chromosome located? How about maybe we should start with that. In the cytoplasm, right? We have a defined region known as the nucleoid, but it's in the cytoplasm. So in prokaryotes, replication takes place in the cytoplasm. Transcription? Same exact location. Translation? Cytoplasm. Okay? What is the importance of this? Well, the importance of this, there's a couple of features. One, we can actually have transcription and translation on an mRNA occurring at the exact same time in a prokaryote. That never happens in a eukaryotic cell. The second thing, and we'll come back to this later on, is it means that prokaryotic mRNAs are actually different looking than eukaryotic mRNAs. So things like introns and exons that exist in eukaryotic mRNAs, we never find those in prokaryotes. Every single region of an mRNA is considered to be an exon. The other thing that this allows for is it allows for bacteria to adapt to changing environments really quickly. So whereas in a eukaryotic cell it might take a few minutes from an initial signal to activate transcription, and it might take a few minutes from transcription to process the mature mRNA, and then to export that mRNA for translation. Talking maybe 15, 20 minutes, roughly, maybe more. In a prokaryotic cell, we're talking like a minute for all of these things to take place, because it can occur all at the same time. So the lack of spatial separation between these different events, what that allows for then is for prokaryotes to change gene expression and to activate transcription, replication, blah, 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 very, very quickly. I just want to kind of show you what this looks like. So this is an electron micrograph of a prokaryotic cell. A couple of the features we'll talk about later on, but here is the nucleoid. And we can define it because it stains differently than the rest of the cytoplasm. Okay, so that's where our events of transcription and replication are taking place. What are these other features? What's this out here? What could this be? Maybe we'll come back to this on Wednesday. Okay. But start to think about what are these other structures out here too. All right. And I did want to point out the pilus real quick because I did bring this up. So the pilus, again, is this attachment protein helping to attach two cells to each other. This one actually not only has the pilus for attachment, but you can see the fimbriae kind of lined up on the outside here. The pilus is hollow, which leads to the difference in staining here compared to the fimbriae. And that hollow tube, almost like a straw, allows for the genomic material to be passed between cells. Okay. All right, let's talk morphology, shape. What are the major types of bacterial shape? Spheres, rods, spirals. And of these three, which is your favorite macaroni? Spiral should be the correct answer. Okay, spiral mac and cheese is the best mac and cheese. All right. Spheres, we also call them coccus or cocci. Rods, we'll call bacillus, and then spirals, spirillum. There's actually another term called spirochetes, and we'll make a distinction between the two of them. We also have additional prefixes that we can add to these names to help describe these a bit more. So for example, Staphylococcus aureus. The name tells us about the morphology of the organism. It tells us that at least they are circular. What does the staphylo tell us? Anybody know what Staphylococcus aureus looks like under a microscope? I think you guys actually looked at this on Thursday. Mancini? It is the cluster. So staphy or staphylo means cluster. Staph aureus is often described under the microscope as having this grape-like clustering. Okay. Another type, strepto. Jake had strep throat the other day, caused by Streptococcus pyogenes. Streptococcus, again, spherical. What does the strepto tell us? Chain or strip, right? So a Streptococcus bacterium has a chain of these strepto are these cocci cells with each other. Okay. Diplo, 2. All right. Bacillus, we can find similar morphologies. Diplobacillus would be just 2 bacilli. Streptobacillus, chain. Okay. Sometimes bacterial genus names can tell us about their shape. So for example, Bacillus cereus is an organism that causes foodborne illness. But the name, the genus name there, Bacillus, tells us about the morphology of the organism. Okay. And we see this again and again. I've already brought up the example of Staphylococcus and Streptococcus. We also see it with others. Lactobacillus tells us the shape is rod. Lacto tells us something about its method or what it produces. And we'll come back to those guys a little bit later. There are a couple of different flavors of spirals, or spirillum. There are ones that kind of fall somewhere between the rod and the spiral, known as the vibrio, which are oftentimes described as being bean shape. Vibrio cholerae, cause of cholera, fits this kind of shape. Okay, so they've got a little bit of a bend to them. But then there are additional spirals. Spirillum are typically a little bit thicker. Oftentimes we're going to find associated with them flagellum. This is in contrast to spirochetes. Spirochetes oftentimes are thinner. They have more of a greater number of corkscrews than do the spirillum. Spirochetes, typically thinner, but there's another distinction between the two of them. And I think this gets brought up in the mini lecture. What is the distinction between spirochetes and spirillum? And maybe we need to skip ahead. I'm not sure if... There we go. So take a look at this image. Oh, you can't see that because I have not advanced it. Oh, there we go. Here is an image of a spirochete. What's different between this and how we drew the spirillum? And take a minute to talk it over with somebody else before we discuss it together. I shall drink coffee. All right, so what's the difference? How do spirochetes differ from spirillum? Flagella, how is it different? It's inside. It's an endoflagellum. This is not a true flagellum. Okay. Some folks will refer it to, well, I refer to it as an endoflagellum. Basically what there is there are two of these. There's one on each end that kind of meet in the middle. And these corkscrew or move underneath a sheath on the outside of the cell. And as they move, what you're going to observe is this moves exactly like a corkscrew does, through liquid or through medium. Treponema, which is the cause of syphilis, is a spirochete. Okay, let's practice for a few minutes. I'm going to take a break. That's not what I want to do. Here are 4 images of different bacteria. What I would like you to do is describe for me not only the morphology you observe, okay, but also what is the name of the flagellar structure. Now, the one you will not be able to give a flagellar structure for is this one right here. This is a negative stain, like the one that you guys did on Thursday in lab. Negative stains and gram stains, we cannot see flagellum because they're too thin. So we have to use different techniques to be able to detect them. But the other three, you should be able to describe the structure here. All right, I'll give you like 3 minutes. Who would like to draw? Use the tablet. See, this gets like 6-year-olds really excited, guys. That's what I'm used to dealing with, though. Okay. I'll tell you what. If you have questions, yeah, Nancini.
Can you draw it for me?
No.
If I tell you?
Okay, I will draw it for you if you tell me.
Okay, draw an oval. Like, yeah. And lines coming out of both ends.
Like that. Thanks. Great job to you too. Lophotrichus, or I'm sorry, Amphitrichus. It doesn't. The problem here doesn't necessarily tell us is it Amphitrichus and Monotrichus, or if it's Amphitrichus and Lophotrichus. So I'd take either of if you drew just one out of each end, and then the bacillus rod-shaped. Okay. Russian fireball, Pyrococcus furiosus. What are we drawing for this one? Alana? Okay, so we've got a tuft on one end. Can somebody help us with the first part? Sphere, circle. Okay. Pyrococcus furiosus. Not technically a bacteria, archaea, but that's roughly its shape. All right. How do bacteria use these to move? So we've just kind of covered flagellar structure, but how do they actually move? And the short answer is there is some randomness, but there's also some specificity. Okay? Terms that are commonly used to describe bacterial movement are the terms run and tumble. So what do we mean by that? What we mean by run and tumble is that bacteria move towards increasing concentrations of chemoattractants and away from higher concentrations of chemorepellents. The process of movement is known as chemotaxis, movement towards or away from chemicals. So the movement, bacteria only move linearly. It's not like they are like humans where we can move and then all of a sudden kind of turn on a curve. Bacteria can't do that. They only move in singular directions. Okay? And so they have to constantly reorient themselves to move towards higher concentrations. So let me provide an example for you. All right? Let us say that door back there, because it is 1140, I will model what's going to happen if we move out of here in terms of bacterial movement at 1150. Okay? So imagine that I am moving. I'm getting closer, closer, and now I'm getting farther away. And then what does a bacterial cell do? That was its run. Now what does it do? Tumble. It tumbles. So how does it tumble? Well, it just starts moving its flagellum in the opposite direction. Over time, as they orient themselves away from whatever that chemical is. Okay. Chemoattractants would be things like amino acids, nutrients like glucose. Repellents are going to be those that we might consider to be harmful to the cell. High concentrations of hydrogen ions, for example, an acid, might be considered a repellent. And the way that bacteria actually sense this is they've got sensors on the outside of the cell that help them to basically determine what is the local concentration of these individual attractants and repellents. Okay. So based on the direction of the movement of the flagella, that can lead to one of these runs or one of those freak-out tumbles. All right. Let's finish off with one last thing today, and that is an observation. No, that's not it. Oh, there it is. Okay. Here's an observation. Streptococcus pneumoniae is a bacterium. Human pathogen, causes pneumonia, also can cause meningitis. Here are two conditions. Wild type, or the normal bacteria, and then mutants. One that have a change, okay, that are missing the ability to perform a certain function. So let's assume that this mutation is within the genome. We're looking at here the percentage of wild type or mutant cells that are associated with what's known as a neutrophil. What is a neutrophil? Anybody in anatomy and physiology heard of this term? Past or present? How about we ask a different question? What is a white blood cell?
What do you mean?
What is a white blood cell? That's what I mean. What is a white blood cell? What is the function of a white blood cell?
It sends, like, antibodies to fight itch.
Okay, B cells are a type of white blood cell, and yes, they do send antibodies. That's a good specific example. White blood cells are part of the immune system. Neutrophils, they're phagocytes. In other words, they gobble up pathogens. They eat them. Okay? So here is an example, Streptococcus pneumoniae, wild type cells, mutant cells. certain percentage of them associated with neutrophils, you can assume, well, we'll come back to that in a minute. So what I want you to do here is, I want you to take a minute, and from this table, tell me, what do you observe? You don't have to tell me necessarily the results yet, but just tell me, what do you observe in this data? Okay? And then if you know something about neutrophils, what does this mean? That's the next part. So take a minute. All right, what's the observation? Or what do the results? What do we see? Taylor.
Is it talking about the likelihood of white blood cells to reach streptococcus, like the...
Yeah, exactly. So association here just means interaction, right? It could mean that they're binding together, or they are sticking together in some way. So these mutant ones, 79% of the time, they are found in association with neutrophils. The wild type ones, about 40% of the time. 2 times more often in the mutants, we find them associated with neutrophils. What that means, and I'll just kind of give away part of this, what that means is that the mutants are more sensitive. They're going to more often be broken down, gobbled up by neutrophils, phagocytized, and basically kind of a sugar-rich coating around the cell. Kind of like a cloaking device in some ways. In this case here, that glycocalyx makes these little buggers really slippery. It makes it tough for neutrophils to grab onto them. Okay? So the last question up here, what properties does a glycocalyx give Streptococcus pneumoniae? So take a look again at the wild type versus the mutants. Which are more frequently associated with neutrophils? The mutants, right? And again, they're missing glycocalyx. So what does the glycocalyx do? Yeah, that's a great interpretation. Protects the streptococcus, prevents them from being destroyed by neutrophils or from being detected by the immune system. So the glycocalyx is involved not only in helping microorganisms sometimes to attach to surfaces, but it also will allow them to be resistant to the immune response. And we see this with streptococci, we see this with some other bacteria as well. And we'll come back to this feature. Towards the end of the semester, we'll talk about the glycocalyx again and its importance in the immune response. Okay, with that said, that's it for today. As you're heading out, I have a couple of things to hand back. One are the quizzes, and then from last week, and then the homework assignment, or sorry, lab assignment from last week as well. The key for the quizzes, these should be up. So under lab, there's a separate folder now labeled as lab quizzes, and the keys for those are available for you to look at. Might be worthwhile since we have a practical towards the middle of the semester. So after I give you guys back these, you're free to head on out.